This video describes the proline biosynthesis pathway project from the winter of 2014 to 2015. Functional genomics is the benchtop component of a genome analysis project. In functional genomics, one attempts to make an experimental connection between the DNA sequence and its function. In our case, we will use the complementation assay to determine if we can get M-Ruber proline biosynthesis genes to be expressed in an E. coli null strain host. Just a reminder about the proline biosynthesis pathway. This is a metasite pathway for proline biosynthesis. This is a universal pathway found in organisms ranging from bacteria to humans. The first two enzymes, gamma-glutamyl kinase and glutamate 5-semialdehyde dehydrogenase, which is also called glutamyl phosphate reductase, form a multi-enzyme complex. Complex formation is necessary for the activity of glutamyl kinase, which is Pro-B, but not for the enzyme encoded by Pro-A. The product of the complex, L-glutamate 5-semialdehyde, rearranges spontaneously to form 1-pyrroline 5-carboxylate, which is converted to L-proline in a single step, catalyzed by pyrroline 5-carboxylate reductase. The gold coloration indicates that functional evidence in E. coli has been published for all three enzymes. This slide shows the underlying theory of a complementation assay as described on the GD Science website. At step 1, we see the Pro-A+ gene for M-Ruber. This gene is isolated from the other genes in the genome by PCR. At step 2, the M-Ruber Pro-A+ gene is inserted into the PKT1 vector through the process called ligase-independent cloning. The M-Ruber Pro-A+ gene is inserted downstream of a strong promoter region. Consequently, under the right conditions, the M-Ruber Pro-A+ gene will make a functional Pro-A protein product. At step 3, M-Ruber Pro-A+ in PKT1 is transformed into a special type of E. coli called a null strain. This null strain does not have a functional Pro-A+ gene. In other words, it is a Pro-A- auxotroph. The null strain must obtain proline from its environment to survive. However, the null strain can get back its ability to make proline if the M-Ruber Pro-A+ gene has the same function as the E. coli Pro-A gene. In step 4, we test if the E. coli null strain gets back its ability to make proline using the process called complementation. The E. coli null with the M-Ruber Pro-A PKT1 recombinant is grown on medium that does not contain proline. If this cell grows, then the M-Ruber Pro-A+ gene gave E. coli the function it lost. In other words, the M-Ruber Pro-A+ gene complemented the E. coli Pro-A- auxotroph. If the E. coli null strain does not grow, then the M-Ruber gene might not be what we think it is, or some other biological process is interfering in its expression. This image shows complementation data for the Pro-C gene. 3 types of media are used. On the left, we see minimal media. The middle plate shows minimal plus arabinose. On this plate, we would expect to see our E. coli host grow if it has acquired the Pro-C gene, either from E. coli or from M-Ruber. The PKT1 plasmid uses the arabinose induction system to activate the expression of the cloned insert. On the right is minimal plus proline, and this is a control to make sure that all strains do grow when given the end product of the proline biosynthesis pathway. So as we look at these plates, if we look at the left, the minimal, on this plate that we see both the E. coli wild type and the E. coli null strain that has the E. coli Pro-C plus clone have grown. Apparently there is a sufficient basal level of Pro-C gene expression from the E. coli Pro-C PKT1 clone to allow for growth on minimal. In the center plate, on this plate, we see that the M-Ruber Pro-C plus clones do complement E. coli Pro-C minus null strains. This would be samples 5 and 6. And then on minimal plus proline, all our strains grow when supplied with the missing end product of the biosynthesis pathway. There is a complication to the complementation assay for the proline pathway. In bacteria, GK plus GPR form a multi-enzyme complex that is essential for GK function, but not for GPR function. The question arises whether an E. coli glutamyl kinase, GK, can function when it forms a complex with an M-Ruber GPR or vice versa. In the complementation assay performed in this class, this scenario will be tested in two ways. One, an E. coli null strain for Pro-B will be transformed with an M-Ruber Pro-A plus clone. Or two, an E. coli null strain for Pro-A will be transformed with an M-Ruber Pro-B plus clone. This slide is just to remind you that the Pro-B and Pro-A genes are part of an operon in E. coli and probably also in M-Ruber. In anticipation of issues with the interspecies protein interactions between Pro-B and Pro-A described on the previous slide, we also cloned the E. coli and M-Ruber Pro-B-A region into the E. coli Pro-B- and E. coli Pro-A- null strains. In the schematic on this slide, I've tried to show which of the various combinations of Pro-A and Pro-B genes in E. coli null strains in M-Ruber have the potential to complement or not complement. So the left-hand column represents the E. coli null strains, the combinations of Pro-A and Pro-B in the E. coli null strains. Then in the next column is the combination of Pro-A and Pro-B and the various clones that we made. And then the third column is the predicted complementation results. And what we see is that in every line, we have the two null strains. We have Pro-A minus auxotroph. We have the Pro-B minus auxotroph. And in every combination of E. coli and M-Ruber, there is some potential to have an interspecies interaction between the Pro-B and the Pro-A proteins. However, my hypothesis is that if we insert or transform the E. coli Pro-A auxotroph with an M-Ruber Pro-A Pro-B clone, that hopefully the M-Ruber Pro-A Pro-B clone would interact to form a functional Pro-B as well as a functional Pro-A, and that we get complementation. Because that scenario, you wouldn't have to have an E. coli protein interacting with an M-Ruber protein. It could happen, but it wouldn't have to happen. And then I was also hoping, as indicated by the other green box, that in the Pro-B negative auxotroph, so the Pro-B null strain, that was transformed with Pro-A and Pro-B from M-Ruber, that again we would set up the scenario having this non-interspecies interaction between the two proteins. So I anticipated we would not get growth if the M-Ruber clone was only one gene, either the Pro-A or the Pro-B, but I'm hoping that we would get growth in either auxotrophic strain when we transformed it with the Pro-A, Pro-B full region. There's another complication to our complementation assay study. A Shine-Dalgarno sequence has not been predicted within 8 to 13 base pairs of either the start codon of Pro-B or Pro-A in M-Ruber. Without a Shine-Dalgarno sequence like the one identified in front of E. coli Pro-A, this might mean that our Pro-A gene will not be expressed in the Pro-B-A clone of M-Ruber. Here's our data showing the outcome of a complementation assay between the M-Rub Pro-B-A clone, which would be M-Rub 1079 and 1080, transformed into the E. coli Pro-B minus null strain. The top line is one student's data, one set of clones. The middle is another student's data. The third row of plates is a third student's data. As you can see in a typical complementation assay, we have the minimal plus arabinose, and minimal plus proline plates. What we see in these set of plates is that our control of the E. coli Pro-BA clone inserted into the E. coli Pro-B- null strain did complement. That's sample 4. And you can see sample 4 grew actually on both minimal plus and minimal plus arabinose. However, our Amruber Pro-BA clones inside E. coli Pro-B- null strain did not grow. That would be samples 5 and 6. So we clearly have an issue of something preventing the Pro-B and Pro-A clones from being expressed inside the E. coli host strain. So table 4 shows the predicted and actual results of our complementation assay. Column A represents the null strain, the genetic background of the host, the E. coli host. Column 2 identifies the region that we cloned into the PKT1. So in some cases it was the E. coli as a positive control, and in other cases it was some combination of Amruber genes. And then column 3 and 4 describe my predictions and an explanation for either a positive or negative complementation assay. And you'll notice that complications might be this whole interspecies BA complex issue, or this issue of the Pro-A and Amruber not having a Shine-Delgarno sequence. So those are the complications. I'm going to go right down to the bottom of this table. Amruber, what is it? Amruber 1345, what we're predicting is Pro-C, very definitely complemented the E. coli Pro-C null. So we know that Amruber genes can be expressed in E. coli. Then as we look at the other results, we can see that all the positive experiments worked. So E. coli clone was always able to complement the corresponding E. coli null strain. So that means that our experiment, the media was made well, and we had in fact cloned the correct E. coli genes. Now, unfortunately, none of the other Amruber clones complemented the E. coli strain, including the BA clone. Now, actually, this might be exactly what we'd expect to find. If we have both the interspecies BA issue as well as the Shine-Delgarno issue, we would not have expected any of these clones to complement, because as we saw in the previous slide, there's always some combination of interspecies interaction needed, or the whole Pro-BA Shine-Delgarno sequence. While I was originally predicting that maybe the Pro-BA, Amruber Pro-BA clone might complement, in the end I guess I'm not surprised that it didn't. These interspecies interactions between Amruber and E. coli I think are complicating the issue. My goal for future studies is to investigate other strategies for analyzing Amruber Pro-B, Pro-A function. Well, thanks for listening to part 2 of the Amruber Prolene biosynthesis pathway study. Hopefully there will be more videos in this series in the future.